So taking a step back and looking at the entire cell here, we can describe the cytoplasm of the cell, including the names and functions of main organelles. And so they're listed here, and if we were to actually see this cell in real time, what we would see is that this cell is filled with fluid, which is the cytoplasm. Again, made up of mostly water, but with various chemicals dissolved in it. So various ions, sodium, potassium, chloride ions, but also chemicals like proteins, enzymes, nucleic acids, all the chemicals that we saw in the last unit. This video is not going to go through each of the organelles found here, but we're going to look at some of the main organelles, and on top of that you're going to complete the worksheet for this week where you actually go through each of the organelles and look at their structure as well as their function and fill in the worksheet. So let's start here with the mitochondria. Taking a closer look at the mitochondria, it has this structure here where it actually has two membranes. It has an outer membrane and an inner membrane, and then there's all kinds of twists and turns in the inner membrane that make sort of finger-like projections, and we call those cristae. Inside the plasma membrane, there are various enzymes that play a role in the mitochondria's main function for the cell, which is ultimately energy production. So the mitochondria is the power plant of the cell, where the cell actually gets fuel. So it gets things like glucose and so on. Glucose is the main fuel that it uses, but glucose and proteins and fats. And this power plant, the mitochondria, will actually convert that into energy, which is in the chemical form of ATP. Before we get into all the steps involved in the mitochondria and its production of ATP, let's briefly explain the role of ATP in the body. Here's the structure of ATP. ATP stands for adenosine triphosphate. And so, if we break that word down, we see that there's three phosphates, each with high energy chemical bonds, and we'll talk about those in a minute. And those three phosphates are where the portion of the name triphosphate comes from. And those are all bound to the sugar, and then the sugar is bound to a base. The base is actually an adenosine base, just like we saw back when we were talking about DNA. So this is the adenosine triphosphate that carries energy around the body. It carries the energy in terms of these high energy chemical bonds. We can compare that to stored energy in terms of a mousetrap in this case. If you've ever set a mousetrap, you actually extend the arm back. You're actually pressing against the spring-loaded portion of the mousetrap. And so then once a mouse actually steps on the trap, the stored energy that's within that spring is actually released and converted into movement of the trap itself. In a similar vein, the high energy chemical bonds between the phosphate groups give off energy when the ATP is actually broken down. When one of the phosphate groups is released, then energy is released from those chemical bonds and allow processes to occur within the body. So ultimately what's happening, in a similar vein to the stored energy within the mousetrap, is our bodies will take these nutrients that we get from eating and drinking. The mitochondria will convert them into ATP, this form of stored energy that we can then use later for things like running and moving and so on. So let's take a closer look at metabolism and the production of energy. Specifically, we want to be able to differentiate between aerobic and anaerobic metabolism. And so, there are three processes that we ultimately need to look at. Glycolysis, which is a series of reactions that we won't get into too much detail about, but we'll look at what they actually produce. Krebs cycle, which is a cycle of reactions. Again, there's a number of them, and we're not going to look into the details of each chemical reaction that's involved. And then the electron transport chain. So let's start with glycolysis. Glycolysis is a series of reactions that occur within the cytoplasm that take glucose and produce another chemical as the product, pyruvic acid. We'll look at pyruvic acid in just a moment, but one thing we need to know about glycolysis is that the series of reactions that occur here do not require oxygen. and therefore glycolysis occurring in the cytoplasm is an anaerobic form of metabolism. It doesn't require oxygen at all, and so in our cells we can produce energy via this process without any oxygen involved. The amount of energy that we produce is 2 ATP molecules by taking glucose, undergoing the process of glycolysis, and converting it into pyruvic acid. Another chemical that we'll look at that's produced in this series of reactions is NADH. We'll come back to that in just a few moments here, but it's important to know that it is produced. So the next step is that the pyruvic acid will move into the mitochondria, into the matrix, the fluid that is found within the mitochondria itself. And a series of reactions that we'll call the Krebs cycle will take place. In the end, the Krebs cycle will produce 2 more ATP, which is what we need, energy. We'll also produce some NADH, just like we did in glycolysis. Furthermore, we'll produce another chemical called FADH2. Again, we'll look at that in just a moment, but know that it is produced via the Krebs cycle. One thing we should note is that the reactions here, taking the pyruvic acid and going through this cycle, the Krebs cycle, require at certain stages some oxygen as a reactant. And what ends up being produced is carbon dioxide, which ultimately we will breathe off via our respiratory and cardiovascular systems. We'll come to that a lot later in the course, but it's important to note that carbon dioxide is actually being produced here and that oxygen is actually being used as a reactant. Hence, the Krebs cycle is actually aerobic metabolism. It requires oxygen. So our next step here is that the NADH and the FADH2 produced along the various steps here will actually go through a series of reactions that are produced by enzymes that are in the inner membrane of the mitochondria. And the electron transport chain takes these reactants, the NADH and the FADH2, and uses oxygen as a reactant as well, and produces water as a product. So again, the electron transport chain occurs under aerobic conditions when oxygen is present. In the end, ultimately, it produces 32 ATP. That's important to note because you can see that glycolysis produces a little bit of ATP. The Krebs cycle produces a little bit of ATP, but really the electron transport chain is the main source of energy production in our cells. But one other thing that's important to note is that we only produce this ATP in the electron transport chain when we have oxygen under aerobic conditions. We only produce the 2 ATP in the Krebs cycle when we have aerobic conditions, when there's oxygen present. And so under anaerobic conditions, we are very limited to the amount of energy that we actually produce. And so think about a scenario where I go out for a walk and my muscle cells need energy. If I'm going for a walk, I'm getting plenty of oxygen, and so the glucose that I'm going to break down to produce the ATP is going to go through all 3 of these stages. I'll produce 36 ATP. If I start to sprint down the road as fast as possible, my breathing rate is not going to be able to keep up with my requirement for oxygen in my muscles. And so what will happen is, without the oxygen going into the electron transport chain, going into the Krebs cycle, I will not produce nearly as much ATP for my sprint because I'm only using glycolysis to produce ATP. So in the end, as you know, I can only sprint for a short period of time, whereas I can walk for a very long time because I'm producing much more energy. One other aspect we need to consider about anaerobic metabolism is that if the Krebs cycle and electron transport chain are not functioning, then pyruvic acid needs to go somewhere. And so what happens during anaerobic metabolism is that pyruvic acid is converted to lactic acid. This lactic acid will then build up in tissue like your muscles, causing your muscles to become more acidic. This is the reason that, again, you can't sprint for as long as you can run, and can cause more detrimental issues if, for example, this is occurring due to some illness. Under normal conditions, lactic acid, once we move to aerobic metabolism can be reconverted into pyruvic acid, and then the pyruvic acid can enter into the Krebs cycle and the original aerobic metabolism can occur. Okay, so we've been talking about the substances outside of cells and within cells, and we've been talking about them moving around, and we've talked about the plasma membrane itself, but we need to be able to describe the methods by which substances enter and leave cells. Because ultimately, for example, the glucose and the oxygen that we're using in order to produce energy need to get into the cells. We also produce carbon dioxide as a waste, and so we need those to leave the cells, and so how do they do so? So ultimately, we're looking at membrane transport. How do we actually transport these chemicals across a membrane, the plasma membrane? And so there's two main types of transport, passive transport and active transport. We want to compare and contrast these two types of transport. And so passive transport requires no energy, while active transport requires energy. Furthermore, passive transport always moves substances from high concentration to an area of low concentration, where active transport can move materials, move chemicals from an area of low concentration to an area of high concentration. So let's look at an analogy here to make sure we understand this idea of concentration as well as energy. Passive transport, think about it as if you had a ramp with a ball at the top of the ramp. There's high energy gravitational pull of the ball as it's going from a high location to a low location, and so this is passive transport. The ball will roll down the hill and it will require no energy to do so. While active transport, we want to go uphill. We want to go from an area of low concentration to an area of high concentration, and so it would require energy in this case to actually push the ball up the hill. And so the same thing in terms of chemicals. If we want to move more chemicals into the cell from the extracellular fluid, but we already have a lot in the cell, it will require energy. Let's take a closer look at the different types of passive and active transport. There are three subcategories to the passive transport. The first is simple diffusion. Again, because it falls under the passive transport heading, it is requiring no energy, and it is movement from high concentration to low concentration. So let's look at an example of me taking a sugar pill, let's say, and placing it into a beaker of water. What will happen over time, because the water particles are moving around and bumping into the sugar will start to move from the area of high concentration in the pill itself to areas of low concentration. This is the idea of simple diffusion. Except in the case of simple diffusion, we could imagine that outside the cell, there's a lot of sugar. Inside the cell, there's not very much sugar, and the particles will move from the outside to the inside of the cell. Another example would be the smell that a skunk produces. It actually releases chemical into the air, which at first is very concentrated around it, but then it starts to spread throughout the air as the air particles move, and it spreads out throughout the area, and then you actually start to smell the chemical. This would be another example of simple diffusion. An example more related to anatomy would be that of the lungs and the movement of oxygen in and out of the lungs and carbon dioxide in and out of the lungs into blood vessels. So when you take a breath in, you have a large amount of oxygen within the alveoli, within the lungs, and you have lots of blood vessels surrounding the alveoli of the lungs. Because there's a high concentration of oxygen within the alveoli, through the process of simple diffusion, the oxygen will move into the blood vessels. There's a high concentration of carbon dioxide within the blood itself, and so it will move from high concentration in the blood to low concentration within the alveoli, thus allowing us then to breathe out the carbon dioxide. The second passive transport type that we'll look at is facilitated diffusion. Again, this works very similar to simple diffusion in terms of going from high concentration to low concentration, not requiring energy, but in this case, it's facilitated. What that means is it's being helped in some way. And what it's referring to is, let's say we have a particle that is too large to actually move through the plasma membrane like we just saw the oxygen and carbon dioxide doing. then we could have a carrier protein within the plasma membrane. We actually looked at the plasma membrane earlier and we saw that there were various proteins within it. One type of protein is a carrier protein that will actually take in the molecule that needs to be transported, and when that molecule moves into the carrier protein will change shape without any energy required in order to release the particle, let's say, into the cell. Or it could be out of the cell from, again, an area of high concentration to low concentration. Because it requires a protein to help the process of diffusion, we call it facilitated diffusion. The final type of passive transport is osmosis. Osmosis is basically identical to simple diffusion, but it's related to water and water only. So, water will move from areas of high concentration to areas of low concentration. So in our image here, we can see that on side A, we have a dilute solution. There's a little bit of solute, something dissolved in the water. And on side B, we have a high concentration of solution, lots of whatever the chemical is dissolved in the water. The water will move from side A, where there's a lot of water, to side B, where there's not as much water, from high concentration of water to low concentration of water. Water is so important in our body that it actually has its own form of passive transport. Even though it's identical to simple diffusion, it has its own name. What would happen over time here between side A and side B is, as water moved through the selectively permeable membrane, what that means is it's only allowing water through it, the water would move from side A to side B in an attempt to go to all the chemical that is creating this concentrated solution, and in the end side A and side B would have the same concentration because osmosis has occurred. So let's look at a couple examples here of solutions that we might have a cell within. The first is an isotonic solution. What this means is, iso means the same, and tonic is referring to concentration. So if we take a cell and we place it in normal saline, which means the same concentration of salt in water as is inside the cell, we actually have two solutions, the solution inside the cell and the solution outside the cell in the intracellular and the extracellular fluid. And so these are isotonic solutions, the same concentration relative to each other. So water will move into the cell, but water will also move out of the cell in the same ratio as the particles move around. We could have a situation where we take a cell and we place it in pure water. This would be a hypotonic solution outside of the cell relative to a higher concentration inside the cell. Hypo means low, and tonic, again, means concentration. And so in this case, the outside extracellular fluid is pure water. There's nothing dissolved into it. And so water will move from the area where there's a lot of water, and it will move into the cell, attracted to the particles that are dissolved within the cell. And so in this case, the cell will start to grow and expand, and then potentially eventually burst. Our final term here is hypertonic, where we actually place the cell into a highly concentrated salt solution. And so what happens here, hyper means, in this case, high, and tonic again means concentration. A high concentration solution outside of the cell relative to the inside of the cell. And so water will move from the inside of the cell to the hypertonic portion, extracellular fluid, attracted to all the particles outside of the cell. These two terms, hypotonic and hypertonic, are relative to each other. So in the example here where we looked at hypotonic, we would say that the extracellular fluid is hypotonic and the intracellular fluid is hypertonic. In the second example here, where the concentrated salt solution was hypertonic, it was relative to the inside of the cell, the cytoplasm, that was hypotonic. Water will always move from the hypotonic region to the hypertonic region. And as we can see in this second example, because water is moving out of the cell, there will be a shriveling of the cell as water is lost. Let's take a look then at active transport. There are two forms of active transport. The first are pumps. Here's an example of a protein in green that's inside the plasma membrane of a cell. This is a pump, and we'll talk about this pump later on. It's a sodium-potassium pump, but this pump actually requires ATP in order to move the sodium from inside the cell to outside of the cell from an area of low concentration to high concentration. In the sodium-potassium pump, it actually pumps potassium from outside the cell to inside the cell at the exact same time, and so hence the name sodium-potassium pump. But the key here is we're going from low concentration to high concentration, and energy, ATP, is required. Another form of active transport are endocytosis and exocytosis. Basically, these two things mean the opposite to each other. So endocytosis is when a cell's plasma membrane envelops some of the extracellular fluid, as we can see here. The plasma membrane invaginates, and some of the chemical from outside of the cell is brought into the cell as the plasma membrane forms around it, forming a vesicle. It brings this vesicle into the cytoplasm to go around the cell and allow for various reactions to occur with the chemicals that it brought in. This is endocytosis. Exocytosis is the opposite. We may have a vesicle with chemicals that the cell has produced, for example, hormones, and the vesicle comes to the plasma membrane, and it actually attaches to the plasma membrane, opens up, and releases the chemical, the hormone, or whatever the chemical is, to the extracellular fluid. This process, again, requires energy. It's a form of bulk transport. Lots of chemical can be transported in or out all at the same time, but it's from an area of low concentration to high concentration. One little trick for remembering endo versus exocytosis is, remember, for the exocytosis, it's referring to exiting. It's exiting the cell. Cyto refers to cell, exo is referring to exiting, versus endo, which means to bring in. Another important method that there is movement of fluids and chemicals within our body is the idea of filtration. And so we'll use an example here of a syringe where there's force on the plunger, there's one area for the water to actually go out the needle, and so the water, as you push on the plunger, moves out the needle. But what if we actually had syringe 2 here, where we had holes in the side of the needle? As we started to push on the plunger and apply a force to it, some of the water would leave the needle, but we would actually have water leaving through the gaps within the needle itself. This is the idea of filtration. We would actually allow some water to press through those spaces. And so this actually happens in our bodies. As our heart beats and pushes blood through our blood vessels, we can actually have water leak out of our capillaries, out of our blood vessels, into the tissue space around our capillaries and blood vessels. Water can leave, other chemicals can leave as well, and we'll look more at that when we get to the cardiovascular system later in the course. But this is an important aspect to realize, that we can actually filter, for example, the blood, where water and other chemicals leave, but as you can tell here, the blood cells are actually too big to leave out through the cells that make up the capillaries, make up the blood vessels. So filtration is another important method that we should know in terms of substances entering and leaving tissues. So another important part of the cell that we need to know about is the DNA. And so let's look at the composition, location, and function of DNA within the cell. DNA is found within the nucleus of the cell. We've looked at it a little bit already in terms of the bases and the sugar phosphate backbone that make up the DNA. So it's important to remember that content that we've already discussed. But we also need to look at the function of DNA within the cell. Ultimately, DNA is the genetic code for how to build all the proteins within our body and within the cell. And so ultimately, we make proteins out of this genetic code, the DNA, and we do that via the processes of transcription and translation. Transcription is when we take DNA and it unwinds and we make a copy of the DNA from another type of nucleic acid, which is RNA. Specifically, as you can see in the image here, we make mRNA or messenger RNA via the process of transcription. mRNA has bases very similar to DNA. However, unlike the T's, the thymine that we saw in DNA, the RNA actually has uracil, a different nucleic acid. But the matching is very similar in terms of the DNA and the RNA, just like it was for the DNA itself. The A's of the DNA match with the U's of the RNA, and vice versa. The G's and the C's of DNA match with the RNA, and vice versa. And so we undergo the process of transcription. And so if we think of a bit of analogy here, the DNA is the code that makes all of our proteins. If you will, it's the recipe book for how to create all of the different proteins that are within our body and within our cells. And so the DNA being the recipe book, the mRNA is more like a genetic copy, a photocopy, if you will, of the recipe of how to make a specific protein. And so what's going on here is this process of transcription or transcribing, making a copy of the DNA into mRNA so that we can take that RNA, that mRNA leaves the nucleus and travels into the cytoplasm and then will attach to this other organelle, the ribosome. This begins the second process, translation. The ribosome attaches to the mRNA, and then what we have within the cytoplasm are tRNA, transfer RNA. Transfer RNA have amino acids bound to them. So there are 20 amino acids that we mentioned in the last unit. So the tRNA actually have specific nucleotide sequences. You can see the 3 nucleotide bases sticking off the bottom of the tRNA in this image here. And depending on the sequence that they have, different amino acids will be bound to those tRNA. In other words, whatever code the tRNA actually has sticking off the bottom in terms of nucleic acid will always relate to a specific amino acid. So what will happen here is the tRNA will actually move to the ribosome. And if the sequence of the 3 nucleic acids that are sticking off the bottom of the tRNA are actually matching up to the 3 base sequence that are coming off of the mRNA, that amino acid that's bound to the tRNA will be added to the growing protein, because ultimately proteins are made out of amino acids, as we discussed in the last unit. Before moving on, we should talk about what these 3 nucleic acids are actually called. On the mRNA, there is the code for which tRNA should come in and therefore which amino acid should be brought in to the growing protein. And so what we call the 3 nucleic acids that are on the mRNA representing that amino acid is a codon. And so as the ribosome moves across, it reads the mRNA 3 base pairs at a time or 1 codon at a time. The tRNA that has the opposite sequence that matches to that codon has a term, the anticodon. And so in the tRNA here, with the 3 nucleic acids sticking off the bottom, this is actually called the anticodon. It matches with the codon. So you can see here that one tRNA with alanine has come in. The CGU has matched with the GCA in the mRNA. And the second tRNA is coming into the second site on the ribosome, and its ACG nucleotides will actually match with the bases, the UGC, found on the mRNA. And so cytidine will be the next amino acid that is added to the growing polypeptide chain. As this process continues to occur over and over, we actually start to form a complete protein made up of the many amino acids that make up the protein. These proteins that are formed may remain within the cell itself. Maybe they're some of those structural proteins in the plasma membrane. Maybe they are enzymes that cause reactions within the cells. But some of them actually might be transported to the surface of the cell and released to cause other functions within the body. And so this brings us back to describing methods by which substances enter and leave cells. One of the methods that substances enter and leave cells, as we mentioned earlier, was exocytosis. And so what we can see here is that perhaps in the nucleus, we are transcribing a gene, creating an mRNA that is coming out of the nucleus into the cytoplasm and binding with one of the red ribosomes that you see lining the rough endoplasmic reticulum here. As the protein is being produced, as translation is occurring, what happens is that protein might be taken into a vesicle that forms from the rough endoplasmic reticulum membrane. That vesicle might move to the Golgi apparatus, where further processing can occur with the protein that's being produced. Perhaps we add carbohydrate to the protein. But then vesicles may pinch off of the Golgi apparatus, move to the surface to the cell membrane, combine with the cell membrane, and open up, releasing this protein outside the cell itself, undergoing this process, as we saw, exocytosis.